Ik zie dat er niet veel vragen bij zijn gekomen. 2 of zo.
Nee, maar we zullen het wel horen hoe het
Ja.
vandaag gaat. Ik bedoel, het is ook een beetje een geste naar iedereen toe.
Ging het gisteren nog goed met je eigen vragen? Ik bedoel,
Ja, op zich prima.
lukte dat een beetje en hosten en vragen?
Nou, is wel lastig, want ik kan niet nog een keer erbij gaan typen of zo.
Nee.
Goed. Even kijken. Nou, mooi. Ik zet... we zijn te zien op YouTube. Heel leuk. Niemand die daar naar kijkt, maar ja, dat is prima. Het zijn toch vaak specifieke studenten die dit soort dingen graag doen. Juist de goed voorbereide lui.
Ja.
En daar doe je het ook graag voor. Oké.
Ik zie dat het bij jou al 1 uur is.
Nee, ik word gek van het geluid van dat ding. Want soms als ik last van mijn longen heb of zo, of verkouden ben, dan slaap ik hier in de logeerkamer. En dan kan ik niet slapen van de klok die tik, 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 tik doet. Dus een beetje kinderachtig, maar daarom...
Maar haal hem dan gewoon weg. Ja.
Even kijken hoor, even de opstelling maken. Dat ik alles...
Opstelling.
Ja,
3-3-2.
2 monitors en dat soort dingen.
Dat is op de chat.
Zo heb ik YouTube in de gaten. Die nog weg.
Er zijn kinderen hoorbaar.
Ja, ik weet het.
Ja.
Het is, ze zitten nu in een terrorleeftijd. Dus,
gaan ze al naar school?
nee, ze zijn 2,5. Maar ze gaan binnenkort weer naar de kinderopvang. Maar dan zegt ze, ik wil dit. En dan zeg ik, nee, dat mag niet. Nee, mag ik dit? Nee, dat mag niet. Ja, maar ik wil het! En dan zeg ik, nee, het mag nog steeds niet. Papa, je moet!
Ja, terrible twos heet dat, ja.
Ja, precies. Maar het aardige is dat ze het in eerste instantie nog op de beleefde manier proberen, maar het ontspoort vrij snel.
Ja.
Maar ja, even kijken. Oké, dat is goed. Nog even de YouTube-link op Canvas knallen voor wie dat wil. Oké, goed. Toch denk ik dat als je volledig zou zoomen, dat het op zich ook wel goed zou kunnen werken hoor. In plaats van fysieke colleges.
Oh.
Ja.
Ja.
Ik denk dat het kan best wel iets hebben, denk ik. Maar... Save. Oké. Nou,
ja, vooral als je het dan ook opneemt of inderdaad op YouTube zo. Dan kunnen mensen alweer daarna afkijken.
Ja, maar dit Zoom kan je dus recorden en dan kan je achteraf alles omzetten in een videostream en een losse audiostream. Dus dat betekent dat je gewoon allebei de dingen je kan het los posten. Dus dat is op zich wel aardig.
Je kan het ook editen?
Volgens mij kan dat wel, ja. Het is gewoon een MP4 die eruit komt, dus die kan je daarna weer editen.
Ja. Altijd gedoe met die collegezalen.
Ja, nou, ik vind het op zich wel leuk hoor, zo'n grote zaal, als iedereen een beetje enthousiast is. Maar dit werkt op zich ook prima. En je hebt hier zoiets, dat heet breakout rooms. Dus dan kan je zeggen, ik heb 20 studenten, ik maak 4 breakout rooms. En dan klik je erop en dan schuift iedereen naar een aparte sessie. En dan kunnen ze daar bij wijze van spreken 10 minuten iets voorbereiden. En dan kan je ze weer terugvragen. Dus je kan wel op een hele andere manier, als je een soort hoorcollege, werkcollege combinatie maakt, dan kan je dat, dan kan je daar best wel leuke dingen mee doen.
De studenten kunnen dan natuurlijk ook weer presentaties geven en zo, neem ik aan.
Ja. Maar je zou zelfs parallelsessies kunnen hebben, dat er verschillende presentaties in de verschillende sessies zijn. Dus op zichzelf, er was toen sprake van dat er een soort collegezaal met 1000 man of 1200 man op Routers Eiland zou komen. Ja. Ik weet niet wie dat precies had bedacht, maar stel dat je je colleges en je werkcolleges gaat integreren, dan zijn dit soort dingen natuurlijk wel interessant. Want dan heb je bij wijze van spreken dat je een kwartier of een half uur hoorcollege geeft en dat het dan switcht naar een werkcollege en dat je dan weer doorgaat als hoorcollege. Ja. Dus heb je wel wat extra mensen bij nodig, maar dan kan je het echt volledig integreren. We hebben nu 7 mensen die wachten in de waiting room.
Ja.
Dus, goed, ik heb hier een chat open. Ik heb de vragen.
Ja, ik heb hier ook het nog eens open. Ja, ik heb ze al beantwoord via Canva.
Ja, dat is prima. Ja. Color blindness. Ik vond het wel gaaf dat, ik weet niet of je dat had gehoord van die mensen die niet stereo kunnen zien. En dat zo iemand toen een keer naar zo'n 3D-film was geweest. Was volgens mij ook een visueel onderzoeker. En dat opeens toen klik zag je het in 3D in de bioscoop. Oh. En toen is hij daar een heel trainingsprogramma mee begonnen. Dat misschien zou kunnen helpen bij mensen die niet 3D kunnen zien. En dat is best wel grappig.
De meeste mensen die geen 3D kunnen zien hebben toch een lui oog of zo?
Ja, precies. Maar ik bedoel, je kan dat dus, als je dat met zo'n virtual reality bril of iets dergelijks doet, dan dwing je het een beetje af. En dan als je dat lang genoeg lijkt te doen, dan lijkt het opeens... Want die virtual reality en bioscoopdingen zijn toch meer 3D dan we eigenlijk normaal gesproken zien. Dus het is een beetje overstimulatie. Oké, ik ga ze erin gooien. Dus we gaan beginnen.
Ja. Ja, ik zag ook echt dat je op die manier zelfs ook nog bij volwassenen lui ook wel weer beter kan krijgen.
Ja, precies. Admit all. Okay, good morning everyone. We're about to start. Again, this is the final Q&A session before the partial exam. If you would have any questions for Victor still, most of the questions have already been answered. Please unmute yourself if you want to ask the question. You can also use the chat and then I will voice it over for Victor. In any case, well, let's start.
Okay, so I have 2 new questions on Canvas. The first one was by Marloes Meijer. I once read that there are special glasses that can help colorblind people see color. How does that work? Well, short answer is probably they don't. As I stated in the answer, I haven't seen or studied these things myself, but of course I did see the internet movies. I've discussed it a bit with other vision scientists and we largely consider it to be a hoax. What could happen, which is sort of similar to what would happen if you put on sunglasses or something, is that the spectrum of colors that reaches your eyes shifts a bit. So that all colors shift to either the more lower or higher wavelength. So that they now see color contrast, for example, orange versus green a bit better because it's now, for example, yellow versus blue or something like that. But it's hardly believable that would do a lot. Another thing which is important to note that this, of course, then would only work for retinal color blindness, where you miss one of your cones. And with retinal color blindness, you still see color, just differently than we do. And you're not able to see certain color differences, such as orange versus green, that we do see. By the way, birds, for example, some birds have more cones than we do, so they can see differences between colors that we do not see. So maybe a bit difficult to imagine, but there are color differences out there that you don't see the difference between, but other animals do. And of course, there are also animals who only have 2 types of color cones, 3 types to be more precise, such as
I
dogs.
understood that the red cone is coded by the X chromosome, so women sometimes have 2 different red cones.
Yeah, that's another thing which is a bit controversial also, whether that's really true or not. But anyhow, so there are colors out there in the world which you don't see. And for example, if you have a dog, of course, a dog sees much less color differences than we do. But of course, the dog also sees in color, just a different kind of color world. So whether these glasses solve that, I'm really doubtful of that. But another thing which is important, it would certainly not work for cortical color blindness, which is achromatopsia, because then you simply cannot see color because you miss the brain area to see color. So any shift in spectrum or what have you, of course, would not help that. All right, Marloes. I don't know whether Marloes is in the room. I think so. Okay. Anyhow, so that was that question. Then there is a second question. I wanted to ask, what exactly do the M cells and the P cells do in the LGN? Short answer again is not very much. It's, apart from relaying information from the retina to the cortex. What happens is that the axons of the retinal ganglion cells contact the LGN cells, and these cells then send their axons to the visual cortex, layer 4, where they enter the visual cortex. The P and M cells in the LGN receive input from different retinal ganglion cells, the midget and paracell cells respectively, but you don't have to know that for now. They also convey different types of visual information. One is color selective, the other one's not, etc. But you don't have to know that either for now. It's not all that well known what these LGN cells really do in a functional sense, other than just relaying the signal from retina to cortex. They may impose some sort of filter in that they're able to shut down this transfer from retina to cortex, for example, when you are asleep or anesthetized or something like that. And of course, all sensory modalities go through such a relay station in the thalamus. Your ears, your senses, your somatosensory information, that all is relayed to the cortex via the thalamus, and the LGN, in fact, is also a part of the thalamus. So those are the 2 questions I see on the on Canvas.
I think there's one additional one. Could you please explain again what retinocortical expansion means? V1, slide 16.
Okay, let me get that. Where are the I think it's this one. Okay, is this one shared now?
Yeah, I can see it.
Okay. Retinocortical expansion. Where is that?
Slide 15, okay. Yeah.
Oh, okay, yeah. 16, yeah, there we go. Okay. It's illustrated here. Here we have the visual field, the left half visual field. And here you have what's called the macula, so that's the center of your visual field. And if you look at the amount of cortex that's devoted to this part of your visual field, it's much larger you can see here. Then the amount of cortex that's devoted to this more parafoveal or more peripheral part of your visual field is shown here. And that's only this part of the visual cortex. So that means that your central vision has much more cortex devoted to it, which is of course the reason why you see better in the center of your visual field and the periphery of your visual field. And this is what we call retinocortical expansion. So the same part of your retina, when it's a central part of your retina, has much more cortex devoted to it than the peripheral part of your retina or of your visual field, which is the sort of equivalent, of course. So that's what we call retinocortical expansion. Is that clear? So where's the person who...
Very clear, thank you. I see here.
Okay.
And another question is: can achromatopsia be inherited?
No, achromatopsia typically is caused by a lesion to the brain, so a stroke or something like that. So it's typically not inherited, no. What is inherited is retinal color blindness, or at least it's a genetic defect. Yeah. Other questions?
I think, well, probably there's people coming into the meeting room, so we definitely have some more questions upcoming. Again, you can use the chat or you can unmute yourself to ask a specific question. Right now, we don't have any questions left for Victor, so please, if you have a question, unmute yourself.
I have a question on the role of P cells and M cells. Do P cells code for color, right? And M cells code for something else? Or is it, or am I wrong?
No, what you're saying is, strictly speaking, perfectly right. The P cells, or at least P cells in the LGN, they are the only cells that convey color contrast information because they get input from the retinal ganglion cells of the midget type, which are also the only cells that convey color contrast information. And indeed, the M cells in the LGN convey, well, other information in the sense that they're not colorblind. There are also other differences between P and M cells, for example, in the amount of detail of information that they convey, spatial frequencies, the temporal way in which they convey information. But those are all things that are not part of the substance well, for this exam. So maybe some other times. But there are other differences between P and M cells. But one of the major ones indeed is that the P cells of the LGN convey color information, whereas the M cells do not.
Another question that was the question was do M cells mostly project to the dorsal pathway and P cells to the ventral pathway?
Yeah, that's also true. Indeed, the P cells primarily project to, well, particular parts of particular layers of V1, and from there to particular, there's this blob and interblob and thinner thick stripe segregation in V2, and then they project to V4, and from there to inferotemporal cortex. So that's ventral stream. This is all lined out more precisely. Let me see. You can see that, for example, here. Yeah, so this is the P cells project to these layers of V1, 4C beta, and then from there to 4A blobs, but probably, and then to the parts of V2, and then from there to V4 temporal cortex. Of course, this is where the majority of the projections go to. Of course, there are also, it's not as strict, of course, as is depicted here. There are also sort of cross connections as well. But then, and the M cells project to a different layer in V1, 4C alpha, and then from that to 4B. Areas like V3, thick stripes in V2, areas like MT, and from there to parietal cortex. So this is a very broad scheme of how these different inputs from M and P cells remain segregated through the visual cortex, and indeed from the most dominant input of dorsal stream, which is the red, depicted in red here, and ventral stream, which is here depicted in blue.
It's not an or non division, right?
No, it's not. It's not perfect, and it's also still a bit controversial whether this is indeed as strict as shown here. But this is the general idea of how these connections seem to be laid out. Yeah.
Okay. Thanks.
By the way, this is the what you see here. So about these different layers where P and M cells project to, and these different blobs and interblobs and thin stripes, interstripes. You don't have to know that for the exam.
Okay. What is meant by cytoarchitectonics?
Is that in reference to a particular slide or something? Because probably not in here. What is it?
Daphne, could you say or chat, is this a specific slide or somewhere in the book that you refer to?
Well, let me answer I'm looking for an appropriate slide. It's not in here. Wait a second. Let me see.
I forgot the slide. It's in V1. Let me check. So Daphne will come back to this. Let's progress to the next question in the meantime.
Mm-hmm.
Yasmina asked, is the visual processing system more of a hierarchical system or rather an analytical process like divide and conquer?
Wow. Well, it's clearly hierarchical in the sense that if you look at the receptive field tuning properties, you know, like orientation selectivity or motion selectivity, direction of motion selectivity or color selectivity or shape selectivities, things like that, you see that they get increasingly more complex, these receptive field tuning properties. Increasingly more high level, you might say, when you proceed from a primary visual cortex to next higher level visual areas. So in that sense, so if we would, for example, look at this slide again. No, this one. So receptive field tuning properties in an area like this one, more appropriate one, maybe not. Wait a second. Yeah, so this is probably the best way to talk about this hierarchy. And so here we have you have primary visual cortex, and then you have all these connections to all these higher level visual areas and even more higher level visual areas. Receptive field tuning properties get more complex and more difficult to understand also if you go to successively higher areas. So in that sense, it's hierarchical. Yeah, so that's why we talk about hierarchical processing. And so the simplest hierarchy, of course, is seen in when you look at what happens when you go from the cells in the retina, which just are sensitive to contrast, to cells in primary visual cortex, which are also sensitive to contrast, but only contrast which has a specific orientation. And so that's a more complex contrast selectivity. It has to be contrast, but it also has to be a particular contrast of a particular orientation. And then if you proceed to even higher visual areas, it, for example, has to be contrast with a specific shape or anything like that. So that in that sense, it's clearly hierarchical. Now I forgot what the other option was. Something about analytical. Can you restate the question?
Yeah, the question was: is it is it a hierarchical system, or more an analytical process, divide and conquer?
To be honest, I don't know what you mean by that.
So Yasmina, could you maybe unmute yourself and clarify?
Yeah, just in the book, it talks about an analytical process, another hypothesis other than the hierarchical one. And it says like divide and conquer. So like one set of neurons focuses on color and then another one focuses on shape, but it's not really, it doesn't really get gradually more complex. It's just all of them combined actually.
Okay. Yeah. Okay. So that's, thank you. So that's in the context of, how in the end a particular percept, particularly a conscious percept, but for example also just recognizing an object, is mediated by these different cells in all these different areas. The classic idea, you might say, is that you have all these hierarchical receptive field properties which get increasingly more complex, so that in the end you have some cell high up which is, for example, selective to you seeing a face. And of course, we know that these things sort of exist in the sense that you have face selective cells. So those are support for this hierarchical idea. But we also know at the same time that for almost any other object, there are no such cells, in the sense that you don't have cells for, say, you seeing a computer screen, or you don't have cells for you seeing a table, or you seeing a dog. You don't have those very specific cells for any object that exists. So somehow, you recognizing these kinds of objects, you consciously perceiving those kinds of objects, has to rely on the combined activation of all sorts of different cells which encode properties of those objects. And so, for example, you seeing, as I'm now seeing myself, a vase with some roses in it. Yeah, of course, I don't have a vase with roses cells in my head. So this is probably mediated by something like, some cells encoding the shape of that object, some cells encoding the color of the red color of the roses, some other cells encoding the shape of the shapes of the leaves, and maybe some other cells encoding the motion because they're moving a little bit in the wind, So, and all these features combined are then forming what is called an ensemble of activity, which is together encoding that particular object. So those are two contrasting ideas of how objects, and also, of course, conscious percepts, are encoded in the brain. The one being, okay, you have all this hierarchical processing, and then in the end, you have cells which are encoding a particular object. Or, on the other hand, that you have indeed divide and conquer. I don't know whether you should call it something like that, but that any particular object is encoded by a whole ensemble of neurons which are coding different properties of such an object. That's the idea there. And in fact, we don't really know yet how this works exactly. It's sort of impossible, of course, to have cells for every potential object that you may see in your life. Of course, that's impossible. So it cannot work in a strictly hierarchical fashion. But on the other hand, we do know, for example, that we have face selective cells, which are encoding very specific object, which is faces. But this probably also only holds for very few, very important kinds of objects, like faces are for us as social animals, and probably for all sorts of other objects. We have to rely on this more divide and conquer or ensemble theory kind of representations. Is that sort of answering the question?
Thank you.
Okay.
Good. So the next question, I think, Daphne, I'm still waiting for the slide about cytoarchitectonics. So I'm continuing with the question. I did not understand the part about how lateral occipital cortex extract shape out of objects that are in motion.
Ah, okay, so let me go there. It's not this one.
It's slide 18 of Vision 2.
Mm-hmm. Oh, it's the wrong one. Slide. Yeah. All right. Well, for one thing, how cells get particular response properties, and so particular feature selectivities, that's largely unknown. But that they have these particular properties, that's a huge field of study. So that ranges from cells in primary visual cortex, which are orientation selective, to in this case, cells in visual area LOC that respond to shape. The previous slide, which is this one, 17, shows you that they do respond to anything that has a shape. For example, they will respond to these kinds of familiar objects, and then you will see activation of LOC. They will also respond to these sort of nonsense objects, but they're clearly objects in the sense that you can see their shape and that they are objects. Maybe you don't recognize them because you've never seen them before, but they are still objects. So LOC also responds to these kinds of things. The only thing the LOC doesn't respond to all that well are these kinds of scrambled objects, because that's just a bunch of lines and features and what have you. But you cannot make any object out of that perceptually. So that's what the what LOC doesn't respond to all that much. That's one thing. But what LOC also does, and that's what this is about, is respond to objects that are defined by what are called second-order cues. Now, maybe I should share the. I don't know whether that works, but anyhow, as shown during the lecture, if these random dots, a region of these random dots move in a particular direction, say to the left, while the other one stays stationary, because you will suddenly see the shape of this object. I think in this case it was a heart or something. It's very familiar to, when you see a butterfly which is sitting on a tree, and as long as it's sitting still, you don't see it, but as soon as it moves, you suddenly see it because of the differences in motion. And then of course you suddenly also see the shape and you recognize it as what it is. And that's what is shown here. So here you see that here you see the response of LOC, fMRI recordings in this case, of course, to different kinds of stimuli. So this is such a stimulus as shown here on the right, but now you can make a giraffe out of it, and then you can see that the arrow is responding to this. This is just motion itself. So motion where you don't see any object emerging. And there you see that the cells don't really respond to that much. This is another OFL stimulus object from, well, I don't know what the L stands for actually now, but anyhow, this is object from it's meant as to be object from motion. So here you can see a dog, and then it responds as well. And here you have an object which is defined just by contrast. So it's just a white rabbit on a black background, and of course LOC also responds to that. So LOC responds to anything that is where you can recognize an object, irrespective of how this object is defined, whether it's defined by contrast, as shown here, or whether it's defined by line drawing, which is of course also contrast, or whether it's defined by differences in motion. And because you can, with these stimuli, very nice stimuli by the way, you can only see the object by virtue of the differences in motion. Because if it's stationary, as you see here, because you don't see anything, you only see the object when it moves. But so this is what we call a second order cue, or in this case, object from motion, contour from motion, the different names to this. But this is also then resulting in you perceiving an object, and apparently LOC is then responding to these kinds of stimuli. Clear?
Is that clear? Yes, it's clear, Bart says, so that's good. Let me check.
Let me go to
There was one question. This is a bit unrelated question to the material itself, but about the partial exam. The question is, how does it work with extra time on the exam? I believe it will be 9 minutes this time, but the education desk will post some additional information on this today. So you have 50 minutes for the clinical psychology exam. Then, so from 1400 to 1450 is clinical psychology. If you have extra time, then you probably have 9 minutes extra. And then 10 minutes rest. Brain and cognition is starting at 15:10, 10 past 3. And then you have 50 minutes to do that. So in that sense, there's always a break to go to the bathroom or at least to get some fresh air, only for just a little amount of time. Okay, good. I think we covered all the questions.
Yeah, let me go to the cytoarchitecture.
Yeah.
Like architecture question. I've taken a slide from some other lectures because that's where it's best explained, I think. So let me share those. So now these are Nissl stains, as they're called, from the cortex, slices of cortex. And Nissl stains primarily color the cell body of neurons. And here you see all the cell bodies of neurons and then going from the superficial layers of the cortex to the deeper layers of the cortex, layers 1 through 6. Now, what this slide is about is that that in different species, the cortex has different thicknesses. But what you can also very clearly see is that these layers, we call these layers because they have there are differences in what's called cytoarchitectonics. If I zoom in a bit.
Composition of different neurons.
Yeah. So that's not very. So if, for example, if you look here, what you see here is that the neurons are a bit bigger. They are a bit clumped together, and here the neurons are a bit smaller, and here the neurons are very densely packed. Here there are no neurons at all; they're just fibers. Here you have all these differences in how what these neurons look like taken together. So the architecture, you might say, of the neurons in all these different layers. Now this anatomical feature, because it's nothing else, but this is just simply something you can see by looking at the cortex in these kinds of stainings. And of course, you also have different stainings where you look at the not at the cell bodies, but at fibers and other things. And so here, for example, you see these columns. And so those are all differences you can see. That's what we call cytoarchitecture. And what Brodmann has done is simply look at the whole brain, at all the different cortical areas, and look to what extent there are differences in cytoarchitecture between different areas. And so here you have different species, but also if you look at different parts of the brain, you will see that in some parts of the brain you have very thick layer 4, and other parts of the brain you have much thinner layer 4. In some parts of the brain you have very large cells in layer 5, for example, in motor cortex you have that. And so there are all sorts of differences in cytoarchitecture, and on the basis of that he has made this division in all these between all these cortical areas, and he just gave them numbers, area 1, area 2, 3, 4, 5, 6, 7, etc. For example, primary visual cortex in Brodmann terminology is called area 17. So that's what we call cytoarchitecture. Now, this doesn't necessarily imply that there are functional differences between these parts of the cortex. It sort of seems logical, but of course, this is just what these areas look like. This is not telling anything about what these cells do. And indeed, if you look at visual cortex, I don't know whether that's in here too. Yeah, so this is actually a better one. So these are all differences in cytoarchitecture. For example, this is in one species. And so here you have area 18. You can see a very prominent layer 3 and 4. Here you have area 17. So here layer 4 looks like this. Here, this is what I talked about, motor cortex, very thick cells in layer 5. So these are very clear differences in cytoarchitecture. And indeed, Brodmann has parcellated the whole brain into all these different cortical areas on the basis of this cytoarchitecture. But as stated here, this is not a functional map. Indeed, if you well, let me put it this way. Oh, sorry. For example, what is here called area 18, that's based on this cytoarchitecture. If you look at it functionally, so what are these cells responding to? What part of the visual field are these cells responding to? Because this is also a visual area. You'll see that area 18 is not the same as, for example, V2. In fact, area 18 is V2 and a bit of V3, maybe even V4. So that's the functional division is not the same as as this anatomical division. But anyhow, this is what cytoarchitecture is like.
Good. I have the response is very clear. Thank you very much. One other question is by Sorina. Can you please explain again which brain areas are involved in view invariant coding?
Ah, okay. Let me go there. This is color motion shape, I think. Yeah. Oh, yeah. Let me share this. Yeah, by the way, these are the slides that go along with the thing I just explained about this analytical versus hierarchical processing idea. So that's also a bit explained here. Okay, viewpoint invariance. By the way, this is an experiment that uses repetition priming. So that's something we discussed yesterday. So that if you present the same stimulus twice to a particular visual area, if that visual area responds to that stimulus, then it will give a smaller response the second time than when it responds the first time. Now, what's invariance? That's the first term you need to know here, of course, which is that you can have the same object like shown here, or you can have the same object as shown here. Of course, these are both umbrellas, but this one is bigger because it's closer by, and this one is smaller. But you can still both recognize them as umbrellas. So in this case, your percept is size invariant. So it doesn't matter what size you see this umbrella, you still recognize it as an umbrella, or a particular cortical area may still encode this as being an umbrella, irrespective of its size. And then it's what's called size invariant. Now, if it would respond also similarly to, say, this chair and this chair, now you will both recognize them as the same chair, but now they're from different viewpoints, and so the viewpoint is different. if the neurons or the area or hue would respond to this in the same way, that neuron or that area or hue would be viewpoint invariant. And then you can also be exemplar invariant. That's not something which is important now, but that's so you both recognize these as chairs, even though they're very different chairs, but you both classify them as chairs. So now you're exemplar invariant. And so it doesn't matter which example of the chair you show, it's all chairs. So that's how we can study these invariances. And that's what they've done here. So the important thing to notice is, again, this repetition priming, so that when you present something for the second time, so repeated object, if the area is selective for that property, it should give a lower response, as shown here. So as you can see here, when you have a particular object and then present it again from the same point of view, you will see that you get this reduction in response. But when it's from a different point of view being, like here, you will see that it will not give a reduction in response. So it's not, in this case, viewpoint invariant. So it is size invariant because that's not mentioned. That's mentioned in the book, but not here in this slide. Because this is the same view, but from a different size, and then it still gives this reduction in response. So it's size invariant coding, but it's not viewpoint invariant coding because there's no repetition suppression when the two objects are shown from different views. So that's what you. So the conclusion is that this area, this part of the brain, has size invariant coding, but no viewpoint invariant coding. Now here you see that the response is also smaller when you present the two objects in different views. That's another part of the brain here on the left. So the conclusion then is that this part of the brain also encodes these objects, but now in a viewpoint invariant way. And so those are the way to study what parts of the brain encode objects either in size invariant fashions or viewpoint invariant fashions. In fact, there is also a region of the brain which is encoding objects in an exemplar invariant way. But that's very close to Broca's area, so that's probably more related to the fact that you both call these things chairs, because what Broca's area is very much related to language. Since you both call these things chairs, of course, this area will then also be exemplar invariant. There's no real visual area which is exemplar invariant.
There's one, I think there's maybe a bit confusion, like here the question is repetition suppression is invariant coding. So that's not the case, but can you explain how you can use repetition suppression to say this is invariant coding or viewpoint invariant coding or not again?
Yeah, I can understand that this is difficult. The whole point of the repetition suppression is that if you present the same stimulus twice, it will respond lower the second time. But of course, then the question is, what exactly is the same? So if you present the exact identical stimulus, so a photocopy of the same stimulus, then of course it's the same in every way. And if a particular area is doing something with objects, or at least with images even, then you would expect repetition suppression for that case, which is of course what you will find throughout visual cortex. Now, let me go back to this one then. If you would present the same object with different sizes, as shown here in B, now from the point of view of a very naive, say, visual area, which is just encoding the pixels in the image like the retina does, you would not expect repetition suppression because they are different in terms of what happens on the screen. And so, of course, that we know that they are the same umbrellas, but a system which is not knowing anything about what these pixels mean, these are just different stimuli. So then you would not expect repetition suppression if this area would just look at pixels and nothing more. But if this area sort of understands that these are the same objects, just with a different size, you would see repetition suppression, because now from the perspective of what these objects are, indeed the stimulus is the same, and then you expect repetition suppression. And so that's what you get here. So they call this same view, but that's what they mean is size invariant. The two stimuli with different sizes. Then so you do get repetition suppression here, which indicates that indeed this part of the brain is recognizing that these are in fact the same objects. So, and that's why it responds this lower the second time. And then similarly here, this part of the brain also responds when they are like this. Yeah, so you have this chair and this chair. Apparently the system recognizes that these are the same chairs because it shows this repetition suppression. And then from that you conclude, okay, it shows this repetition suppression, so it recognizes these are the same chairs. So somehow these objects in this area are encoded in a viewpoint invariant way. So you have viewpoint invariant coding here. Is it making a bit more sense now?
I think there's still some confusion, but I think if you could you go back to the other slide with the ABCD? So there's probably some areas where you show A, that it shows repetition suppression, but in all kinds of higher regions in the cortex not. And then if you show B and then a bigger or a smaller image, then you see repetition suppression in these other areas, but not in the area where it responds to A. So there's basically these different kinds of classes.
No, it's the other way around. So every visual area will of course show, well, pretty much everyone will of course show repetition suppression for A. It's only somewhat higher areas, if you think in this hierarchical model, of course, which will also show repetition suppression for B.
Same object, but size invariant.
Yeah, and it requires even higher, more sophisticated encoding to also show repetition suppression for C. So if anything, you would expect all visual areas to show repetition suppression for A, some of them for B, and even less for C, and even more or less, in other words, for D. Yeah, so because it gets increasingly sophisticated, it requires increasingly sophisticated encoding, of course, to recognize that these are the same, and yeah, of course, it's simpler to recognize that these are the same than that these are the same. Yeah, so if you would go for a hierarchical idea, you'd expect every visual area to show repetition suppression here, and only a few, or maybe even none, for D. Because repetition suppression is showing you that the area is encoding the feature that you are repeating.
Mm-hmm. Is that clear now? Clearer, thanks. I'm just recapping. So A is repetition suppression in most visual areas, B only repetition suppression in size invariant areas, and C only repetition suppression in viewpoint invariant areas. And then of course in D, it's basically a repetition suppression in categorical regions that respond to all kinds of chairs.
Yeah. Yeah, I know it's both the fact that you have to understand repetition suppression and of course this invariance terminology. Yeah, I understand it's a bit difficult, but I would suggest you try to go over it once more, maybe after the way I've explained it now, And if you still don't get it, just drop me an email. Or call me, I don't know, whatever.
Okay. So in that sense, I think these different brain regions have different characteristics. And repetition suppression is basically the technique to see what they're sensitive to. So you can basically say something about lateral occipital complexes viewpoint invariant, for example. And the fusiform phase area shows repetition suppression to all kinds of different phases that you use. So it's basically the category region.
Yeah.
Good. Thanks. Let's see. Do I have any more questions here? I'm just going to go over this. Yeah. Bart asked the question, does the right hemisphere therefore uses a view-dependent frame of reference?
Now this is actually a question about hemispheric specialization, which is something for the next half of the course, where we go into much more detail there. Yeah, there are all sorts of differences between left and right, some of which some are quite controversial and others are maybe a bit more established. I don't think you can simply, on the basis of this one experiment where we've looked at the well, not we, but where people have looked at the viewpoint invariance of these kinds of regions, you can draw such a general conclusion, so that it also always uses a viewpoint, a view-dependent frame of reference or something like that. No, you certainly cannot draw that conclusion. Apparently, in the encoding of these kinds of objects, you see that this part of the brain is a bit more sensitive because, with fMRI, of course, you always also have to deal with things like that. You have to set a certain threshold to see parts of the brain light up and complications like that. So this part of the brain is a bit more viewpoint invariant, and other parts of the brain on the other side are less so. But to draw a conclusion like that would be a bit much.
So the next question is, I think there's still some confusion about apperceptive and integrative agnosia. So they seem quite similar. Could you maybe say what is the distinguishing feature? So how can you dissociate between the two? When is it apperceptive and when is it integrative agnosia?
Yeah. Well, to begin with, indeed, they are sort of extremes on differences in gradation more than really different conditions. And so they're both, the central property of both is this problem of perceptual organization, you might say. And so not being able to make sense of all the different parts that make up a scene, like, this is a well-known example. We sort of readily see that this is a horse with a carriage. But we do so by combining all these blobs into one thing, which is a very robust feature of vision. We always try to make sense of all the different parts of the visual scene. This is what we call perceptual organization. And both apperceptive agnosia, as it says here, is a failure of perceptual organization. And integrative agnosia is in fact the same, and also says it's somewhat similar to apperceptive agnosia. So what's the difference? Yeah, so apperceptive agnosia is just a much more severe case. So in apperceptive agnosia, for example, you will see this deficit in unusual views. You will see this deficit in the shadow test, which may not be that prominently present in integrative agnosia. Because, well, for one thing, it also depends on how many objects you show. If you show one object, integrative agnosia patients typically have much less problems than aperceptive agnosia patients. And so this is a good example of when integrative agnosia patients get into difficulties. You have these multiple overlapping objects where we can still define, see that this is a saw and this is an umbrella and this is a cup and this is a pear, but they really fall apart here because there's so much information overlapping that they cannot distinguish. But if you would show them one saw, many of these patients would still be able to recognize this as a saw.
And a perceptive agnosia would also not recognize one single object.
Exactly. Right? So exactly. So that's, for example, what you see here. These are very severe examples of aperceptive agnosia. There's just one A being shown here, but if the patient is asked to copy it, yeah, you can see it. It just doesn't make any sense anymore. And an integrative agnosia patient would still be able to copy this or recognize this as an A and copy it quite well. What you do see, by the way, with in the copying, that's this example, that integrative agnosia patients do. So the aperceptive agnosia patient is copying like this. So this is just not making, very bad. The integrative agnosia patient can, for example, copy this example as shown here, but he does it in sort of a strange way. Of course, when we would try to copy this, we would just draw a circle and two squares. But this patient just sort of has to follow all the individual lines, as you see here. So it first does this, and then 2, 3, 4, 5, 6, 7, and then it goes like 8, 9, 10, which is of course a fairly strange way to copy this picture, indicating that he's seeing the lines and he's seeing where they're going and he's seeing sort of what the shapes are, but he has difficulty just recognizing, okay, this is a circle and two squares, so he has to do it like this. So that's a big difference between integrative and aperceptive agnosia. If you look at the copying, for example, this is just a mess and this is sort of okay, but the path towards it is a bit clumsy.
Yes, so and is this clear? Okay, very clear. Thanks. Okay. One other question is, Yasmina asks, patients who can recognize orientation but cannot guide their action have optic ataxia. What is it called when patients cannot recognize the orientation but then guide their actions accurately?
Well, that's called optic agnosia. It's of course that's a very general term. This patient, so this famous patient DF of Miller and Goodale, who could not do the matching, so do not perceive the orientation but can do the posting, so can automatically use this orientation to control some action. There's no real name for that except for that they don't visually recognize objects or orientations or whatever. So that's why it's called visual agnosia. Whether that's aperceptive or integrative agnosia, that's sort of, that's not really dealt with here in this literature. But they just have it's probably, this patient probably has something which you could call aperceptive or integrative agnosia. Yeah, so but it's not really specified because it's not really studied on all that detail what exactly the kind of agnosia is in this patient, except for that it's very clear that she cannot make any sense of any visual information. So in that sense, it's very similar to aperceptive or integrative agnosia.
Related question here, does the difference in performance in matching and posting tasks has to do with the movement and position perception?
Well, that's the nice thing about this experiment, because what I did here was that, in fact, all the things that the patient is doing is virtually identical in the sense that in the matching task, of course, the patient is also holding such a, disc or something, that she just has to match the orientation with the orientation of this slot. So, you know, she uses the same muscles, the same thing. She has to do the same sort of task. The only difference is that she's not actually pushing the thing into the slot, so she's not posting it, which is, yeah. So from what happens in motor cortex or from any action planning or what have you, yeah, the two tasks seem almost identical. The only thing being, of course, the only difference being that here you have to, well, do the matching, which is, you know, you have to sort of know the orientation of this slot and then position your hand in the same way. And here you just don't have to, well, you can almost say you don't have to think about it. You can just, you know, let your hand be guided by the visual stimulus, and then suddenly she can do it. So
yeah, so I really love this study, but it's really this dissociation between this ventral system and the dorsal system that you can see here.
Yeah, exactly. So that's because it's using the very same visual information. It's doing almost the very same motor action. The only thing is that here it's much more cognitive, you might say. And so you have to rely on your perception. Here, just do it. You know, just like we do things the whole day, which is very much mediated by dorsal stream indeed.
So was Nike sponsored by the dorsal stream then?
I guess so.
Bad joke. Okay. We're almost out of time. Any last urgent questions for Victor? So I think the question by Yasmina here is optic ataxia dorsal and agnosia ventral. The short answer would be yes.
Yeah, indeed. Yeah. So the optic ataxia patient has a posterior parietal lesion, as it says here. So that's dorsal. Yeah.
Good. Any more questions for Victor? Just waiting a bit. Okay. Please unmute yourself if you have any last questions, else we're going to thank Victor.
Okay.
No, not really, I think. Good. Then thanks, Victor. And
thank you.
Yeah. I'm going to continue afterwards with the other lectures. So please stick around if you want to know a bit more about that. See you soon, Victor. Bye.
Bye-bye.
Okay, so Victor's out. We're going to continue here in terms of managing the chats. Does anybody want to volunteer in co-hosting me here? Because I cannot type in answers to the questions and talk at the same time. So if anybody would like to volunteer, please say so in the chat or unmute yourself.
What are you supposed to type?
A bit of like if I give an answer, then a short version of the answer to the question in the chat.
And put it down in the chat.
Yeah, and also like if there's anything happening in the chat, I won't be able to see it. So basically by saying like this is the question in the chat.
I can do it for you.
Yeah,
yeah.
Claudia, right? Okay.
Yeah.
Make co-host. Yes, thanks. You can keep yourself unmuted. I'm going to start with a couple of questions. I think going to Canvas first. One of the questions is, in which parts of the brain do you see open field versus closed field neurons? I was talking about this mostly in relation to how you can measure EEG. And we covered the topic of cytoarchitectonics a bit just now. So we see this very neatly structured, these open fields where all the pyramidal cells are basically well aligned to each other, mostly in the cortex. So we have these 6 layers of cortex and everything is lined up quite neatly. So if we're talking about these open fields, then we're mostly talking about cortex. And we're not so much talking about the subcortical structure. So that's the distinction here. Okay. So that was the first question that I retrieved from Canvas. If you have any questions about this anymore, thank you, Claudia, for saying this. But open fields, mostly cortex. Closed fields is typically what you find in subcortical structures. I also see a question by Sol here on Canvas. What is the difference between transcranial direct current stimulation and electric convulsive therapy, ECT? I think there's a difference between edition 4 and 5 in the book here. In edition 5, actually transcranial direct current stimulation is treated quite extensively and not in the fourth edition. So if we're talking about TDCS, or as I usually call it, TES, transcranial electrical stimulation, it uses a very low voltage, typically around 9 to 20 volts. So you can use all kinds of batteries that you can buy in store to use that. So it's very low voltage, and the strength of the current is also quite weak. So we're talking about 1 milliamp, maybe 2 milliamps. If we're talking about electric convulsive therapy, you actually use way more current to induce an epileptic seizure, so a controlled epileptic seizure. And this is the main difference between the two. It's the strength of the electrical current that you're using. In essence, it's the same principle. You're just pushing an electrical current through the brain, but the strength of the electrical current is the major difference here. So good. And this epileptic seizure seems to cause a sudden increase in for example, mood in depressed patients. So that's, I think, the clearest thing. TDCS seems to have good effects on depressed patients as well, and you can also use TMS for that. So TMS, sorry, Claudia, TMS is something different. This was ECT, so electric convulsive therapy. Great. And I see a question on slide 43, executive functions regarding the inferior frontal gyrus and filtering. I'm just going to share my screen and open up that part. Let's see. I think this is the correct window. Share. So hopefully you can. I'm going to the executive functions slides. Can you see that?
Yes.
Yes,
Yeah.
okay. I think it's slide 43, right? 43. Yeah, exactly. So what the important thing here is that I was talking about these executive function cycles, basically what happens when you have to show any kind of goal-oriented behavior. You basically need to know what to do, when to do it, how to do it. So you need information in working memory to set the goals and the sub-goals. you have to amplify some information to basically make it the boss in the brain to dominate your response. And in this case, you see a very awkward thing. So whenever you see a stimulus, whenever the context is recruiting different kinds of options for responding, then you actually need inhibition to filter this. And this is clear in this example here. So in the example in the slides, I showed the stimulus of bed. You can basically tidy the bed, you can sleep in the bed, you can do all kinds of other things in a bed. So basically this one stimulus or this one context activates multiple options at the same time. And typically all these options are ready at the same time, but they need to be inhibited before you make a choice and then act out. And this is something that is taken care of by these inhibition circuits. So in all these slides prior to this and past this, I was talking about the inferior frontal gyrus. But even if you show an image to a patient with inferior frontal gyrus damage, and for example, if you ask what can you do with scissors, like I think 99% of the people will say you can cut with scissors. What can you do with a wheel? Then there's actually equally valid answer options. So therefore, you actually have to inhibit the other two in this case to voice one of the options. So you need inhibition to inhibit the multiple options, else patients will really get into trouble here. Is this clear?
Yeah, it's clear. Maybe one question. In real life, is this area always activated because we always have to filter things, or how does that work? For example, like in the gorilla experiment, would be the IFG would be active or not?
There's a whole lot of studies done on this, and most of them are also reported in the book. So the IFG generally suppresses activity in other brain regions. So basically keeping all the alternatives in check. So it has a general inhibition role. And that can be if you are playing baseball and not hitting the ball, but that can also be if there's multiple response options in a verbal task like this. So it has a general inhibition role. So definitely if you're doing experiments and put people in the MRI scanner, even if you don't intend it to, you often see inferior frontal gyrus activation.
Okay, thank you.
Good. So that was one of the other questions. So now, Claudia, is there anything else on the chat that I need to answer?
Yes, there is a question from Iris. Could you please explain the block design and the event-related design again? And can you use both for all methods, or is this method specific?
Okay, good. So I think I have talked about that in my own lectures too. So I think it's, let me go to the right files. Methods of cognitive neuroscience, I think. Probably should be this one. Yeah, good. And I'm skipping to fMRI section. Okay. Did I? Yeah. Good. So what I was, I don't know if you've seen the lecture, but basically, whenever you present the stimulus, when people are inside the MRI machine, you start to see an increase in inflow of oxygen-rich blood. So typically in this image that you can see over here, functional MRI signal from the visual cortex. So here 0 is the onset of the stimulus, and then about 6 seconds later, you see about 1.5–2% change in the bolt signal. So this is basically the response to this one single stimulus. I hope this is clear. If you show a stimulus and then wait, so in this case, A would, for example, be a house, B would be a face. If I show a stimulus A and then wait for a sufficient amount of time, typically around 16 to 20 seconds, then the bolt signal goes back to baseline. If I show the face or a house again, then I see exactly or more or less exactly the same change in bolt signal. The problem here is that if I take this approach, I have to wait for like the full bolt signal to come to rest again before I can present a new stimulus. So this is actually quite inefficient to do because I can actually squeeze in quite more stimuli in time if I want to. And that's something that in the block design actually, come on, don't start loading again. Oh, I clicked on the wrong button. I'm sorry for that. Does somebody remember which slide it was? 57 or something? Okay. So if I, the trick here is, if I squeeze in stimuli and repeat them, say, every 4 seconds, then there is a bit of a return to baseline of the bolt signal. Not entirely, but it doesn't max out. And that's something that I actually want to prevent here. So in a block design, I keep repeating stimuli, stimuli, stimuli. So in this case, I'm repeating a moving stimulus every second I show a moving stimulus, or in this static picture, I keep repeating the same object over and over and over again. So eventually, if a brain area is sensitive to moving or static stimuli, the bolt response cannot be raised any further. So I'm actually maxing out because at a certain point, say like 3 or 4% signal change in the bolt signal, that's the maximum amount that I can increase oxygen-rich blood to this brain area. It simply doesn't fit more. So we don't get this return to baseline. So I cannot see when each of these individual events was presented. And that's something different with the event-related design. So in the event-related design, you actually are jittering the stimuli a bit, and you must prevent at all times that you max out on the bolt response because the stimuli are spaced too close together, because then you cannot estimate back all the individual events anymore. I think that's the crucial part here. So if you're really interested in, say, I want to see which brain region is sensitive to faces or to houses, then you would present lots of faces, and then in a different block you present lots of houses. You're not interested in the response to each individual face or to each individual house, so you're maxing out the bolt signal in both cases. If you do want to see how the response to each individual event is, you either need to space it out by at least 20 seconds, or you have to vary the distance in time. So sometimes it's a phase, and 1 second later again a phase, and then 4 seconds later one phase, as long as the bolt signal doesn't max out. I hope this explains it a bit more.
Yes, thank you. So just to get it clear, the event-related design is really about the event, and the block design is really about the area in the brain.
Exactly, exactly. So the block design is typically not something that you cannot use for any psychological experiment. But we do use block designs, sometimes with repetition suppression, sometimes without, to basically see where are the phase specific and the how specific areas in the brain, where are all the language areas, for example. So you use that to localize specific brain parts in your subject, and then you do the event related design to see how these regions interact or respond in a typical psychological experiment.
And is the event-related design typical for EEG and the block design typical for fMRI, or is it interchangeable?
I think the block design gives you way more signal. So in a very short period of time, you can basically assign meaning, or what does this brain area do. But the event-related event-related design is a nearer thing, and you have to be very smart in how you time your stimuli to not max out on your bolt response, because else you won't be able to retrieve the response to each separate event. So yes, in EEG, you can always use this event-related design. You can also use a block design if you're interested in, say, is there some kind of component in the EEG that is phase selective? So you can still use the block design there. But it's much more common to use block designs in fMRI to really say something about brain specialization areas.
Okay, thank you.
Good.
Yes.
Okay. Next question.
Next question. Do we need to know about the monosynaptic reflex ARC with the afferent inputs and efferent outputs?
And is it specified where this exactly is?
The monosynaptic reflex?
Yeah.
No.
Is this something that only was talked about in the book, or is this also something that was in the lectures?
Yeah, sorry, it was only in the book actually, but it was part of the pages that we were told to read.
Yeah.
And I just took notes about it, the monosynaptic reflex arc with the neural circuits, and I didn't really understand that part, so I was wondering if we have to know it.
I think we're not going to ask you any exam questions on that. I think it's in terms of like understanding this. This monosynaptic kind of reflex arc basically enables you to show all kinds of reflexes without the signal going all the way back to the brain. So if you, for example, well, you grab something and you grab a piece of glass, you immediately have this reflex that goes through your spine, and by one synapse activates your muscles. So actually, your spine on its own can set this reflex arc in motion without your brain being involved. In these more complicated or multisynaptic pathways, for example, you cooked a meal for yourself or for friends, and then you forget to well, you grab the pan from the stove. It's incredibly hot, but you don't want the reflex that you throw the pan away or something like that, because then everything goes to waste. So then you override the pain reflex a bit by just holding on for long enough to put it back on the stove. So we basically have two mechanisms there. So there in the multisynaptic reflex arc, you're actually suppressing the monosynaptic automatic kind of reflex. Okay, I hope that's a bit clearer. We won't be asking you anything of that. I think it's too complicated to ask sophisticated questions, but I think that we have this automatic reflex that basically goes from the pain receptors in your skin to the spinal cord and then directly activates the motor system again. That's a beautiful and very fast design. And that we have some control over this, because if the pen that I was talking about is like super, super hot, you will throw it away immediately or drop it immediately. So it doesn't hold for a very extreme stimuli, but in our cases, you can have some control by basically having the brainstem and the somatosensory cortex implementing some control over the touch that you get. Okay, the next question.
Yeah, another one from Yasmina. Could you please explain mesocortex and allocortex?
Good. Where is this also from the book, or is this Yasmina? Maybe you can unmute and.
Yeah, sorry. I'm just going back to where I saw this.
Mm-hmm.
It's from the same chapter in Brain Anatomy, but it was also from the book.
Yeah. Is it the fourth or the fifth edition?
Fourth edition.
Yeah. Which page?
I'm not sure which page. It was under figure 2.32.
2.32, okay.
And it talks about mesocortex, the paralimbic region, and cingulate gyrus.
Mm-hmm.
And then the allocortex with the 4 layers of neurons.
Mm-hmm.
So what's
right.
Are they 2 different areas of the brain?
So if we're looking here, so the neocortex, well, I don't have to explain that because there you basically see the pink in a picture 2.32. I think in the 5th edition, for those of you who have the 5th edition, the same picture is present there. So just give me 30 seconds to grab that picture too. So I don't know if you have the book in front of you, but this is definitely also in there, almost there. Yeah, that's figure 2.38 in the 5th edition. So in pink is basically the neocortex. If we're looking at the mesocortex in blue, that's basically the limbic system. So that would be another way of calling, like I was talking in my slides mostly about the limbic system. The mesocortex definitely has a different kind of architecture. If we're looking at what cell layers do you see, how many cell layers do you see? So you would definitely conclude that the mesocortex is an older structure in that sense, making part of making up the limbic system. The allocortex is linked to, for example, a regulation of emotions and the amygdala. So that again has a different kind of structure. So I think they're pointing out different structures here, and really making the claim that if you look at the which cell bodies are present, what kind of neurons are present, you see strong differences between these regions. So I think that's the only important message that you have to remember from this. That if you look at different parts of the brain, that this architecture in different layers and different cell bodies is dramatically different. I won't be asking you questions like point out the mesocortex. I would always refer to it as the limbic system here.
Okay. I think that's clear. What's the main difference between MEG and EEG in measuring ERPs?
Good. So MEG and EEG, You can use both to measure event-related potentials. The big difference here is the technique itself. So if we're talking about EEG, you're recording electrical activity. Electrical activity has one problem. It doesn't go through the skull very well. So if we're talking about all these pyramidal cells, they're lying in a gyrus, and on top you have the skull. Like the electrical field goes straight up here, and then it spreads out because it basically needs to find a way to get through the skull. That's also the reason why, spatially speaking, so in space, EEG measurements aren't very accurate because the skull is basically screwing things up. If you would put the electrode directly on the meningi, for example, under the skull, then you won't have this problem. If we're talking about all electrical fields are producing a magnetic field, and this magnetic field basically well, goes through any matter. So it's not barred by the skull, for instance. The only thing is, if we're talking about electrical circuits, so again, if you have all these pyramidal cells pointing up and the skull is here, the electrical field goes in this way, the magnetic field goes in that way. So it's always orthogonal to the electrical field. So if you would, for example, have all these pyramidal cells in a gyrus and the skull is here, you won't measure anything on top with MEG here because the magnetic current goes in this direction. So you need sensors to the sides to measure this. So if we're talking about typically what you would be measuring with EEG, you're most sensitive to pyramidal neurons in the gyri. And actually, if you go a bit to the side, you have this sulci. So all the pyramidal neurons are lying like this. The skull is here. And actually, the magnetic field will point like this. So it's mostly sensitive to whatever is happening in the sulci and not so much in the gyri.
Yes.
Okay. So in essence, you're always measuring the pyramidal cells, the pyramidal neurons in both cases. EEG is more sensitive to gyri, and MEG is more sensitive to sulci.
I think that's clear.
Thanks.
Could you explain the graph with the TMS activation with the two dips? It was in one of the weekly assignments.
Yes, I think so. Let me, then I probably have to go to the weekly assignment, right?
Yeah.
This will take some time. Maybe you can progress with the next question first.
Next question is also about the weekly assignment 2, question 6, and the feedback is also a bit confusing to ST.
Okay. Weekly assignment 2, formative. Weekly assignment. Okay. 10 questions.
Yeah.
Perception. Let me take a look. I'm just going to share my screen again. Okay. Hopefully you can see it now.
Yeah.
This is the weekly assignment 2. So
yeah, the question 6 was first, and
that's not this one, right? This is the dip.
Yeah.
Okay. Okay, so what we're basically doing here is what you see in this graph. People are doing a task where they have to perceive motion. And if we're talking about motion perception, this typically activates area V5 in humans. So that should be clear. And what they do here, so it's actually quite a difficult task. They're applying TMS at different moments in time. So either before the motion stimulus appears or after the motion stimulus appears. And in this case, the final part here, the TMS, no TMS, so this is the baseline condition, people respond about 72% accurate, right?
Mhm.
So what happens here is that if you present the stimulus and then apply TMS before actually, if you, the TMS onset is before the stimulus arrives, then you see that people get worse at the task. If you apply TMS slightly later, so actually after the stimulus has already appeared, then you see this kind of typical late dip. So what would you conclude based on this? Like if you inactivate the region, so to say, before you actually present the stimulus, then this brain region cannot do its job because you basically knocked it out. And once the stimulus arrives at the brain area V5, it's processed less efficiently. Is this clear to everybody? So that's basically you knock it out by means of a virtual region. So when the stimulus first arrives at this brain region, you see a dip in performance. If you present it afterwards, this late dip, if a signal arrives in V5, it's then sent to R regions and then it's sent back to V5 again. So basically, this is an iterative or looping kind of concept. If you target it directly at the right time when it comes back, you get an additional dip in performance. I think.
Okay.
So this is, I think, like, what was the instruction? Oh, sorry, yeah. That's not good. So that's actually what's happening here. If you have any more questions about this, please ask. But this basically shows that if you apply TMS, if you do it at -300 up to say -200, you don't see any effect on performance because V5, you knock it out, but it is not activated by the stimulus yet. If you knock it out just prior to the stimulus, then it's not available for initial processing. If you knock it out after the stimulus has been presented, then these recurring loops of activation cannot interact with V5 anymore.
Okay.
So I think this is, in some sense, a very difficult question. The weekly assignment 4.
Maybe one clarification.
Yeah, sure.
So the second application of the TMS, why then there is a dip also after the onset, after the stimulus onset?
That's actually what they are referring to here, but you don't see it very accurately. So maybe you can see the plus in my magnifying glass here. So it goes to say 67, maybe 66%. So it's actually quite a tiny drop, right? So if you knock it out during initial processing before you're showing the stimulus, so then it drops to 58%. So you basically don't allow V5 to do the initial processing the first time the stimulus arrives. So you effectively knock it out entirely or almost entirely. If you let it pass through V5 the first time, and then it basically comes back to V5, you get a reduction in about like 6-7% in terms of performance. So that's the late dip they're talking about.
Okay.
Okay. I think there were two the same questions about this specific question in the weekly assignment.
Mm-hmm. And what was the other question about the weekly assignment, or was this?
That's the question 6, right?
Yes, I think we were talking about the same question. But
Yeah.
could you please repeat the second deep? I didn't understand the reason for the second deep.
Mm-hmm. So what typically happens in and this is really also related to like conscious perception. So what typically happens if you're seeing visual motion in this case, the signal will go from the eye through V1 to V5, and then to higher brain regions afterwards. But the signal also comes back to V5 after it has been initially processed. And this is something that is quite surprising with TMS. So if you knock out V5 before the initial signal arrives in V5, you get a big decrement in performance. So actually the stimulus itself, the initial time it's being processed, the brain region isn't involved. So motion perception really drops to like 60, maybe 58%. If you then zap it later on, V5 is still available for this initial processing. But if you zap around 150 milliseconds after the stimulus has already been presented, it has been initially processed by V5. It goes to different brain regions, and then it comes back to V5. And when it's then unavailable, you also see a dip in performance, so a dip in conscious perception of visual motion. It's not as strong, but you do see this. So this basically shows that this V5 region is not active once during perception, but actually twice at different points in time.
So both are caused by the initial TMS?
No, because each dot that you see here, you only zap the TMS once. So these are lots and lots and lots of trials. So I zap V5 here around 250 milliseconds before I show the stimulus over and over and over again. Then I try it at 50 milliseconds before stimulus onset, and then you get a very much of a reduction in performance. I zap it around, say, 50 milliseconds after stimulus, and you might even see like an increased performance here instead of a dip. But there's 2 points here where my magnifying glass is over here around minus 100 to minus 50. So before you present the stimulus, you get a big decrement. And when I zap later in a different trial, about 150 milliseconds after onset of the stimulus, I also get a decrement in performance. So this actually suggests that V5 is activated twice and is involved twice in processing motion instead of only once.
Okay, so it was the figure that was confusing.
I think the figure is confusing, and if I'm reading up on this, this is like I would never ask a question like this on the exam because it's too complicated to grasp.
Okay, thank you.
Good. Next question.
Yeah, could you please explain the stop signal reaction time regarding inhibition from the cog control lecture again? I think it's slides 36, 38.
Yeah. Let me go to the slides again. Modules. You can still see my screen or not?
Yes.
I think it's that lecture. Okay. Is it big enough, or do I need to enlarge it even more?
On my screen, it's perfect.
Okay, good. If it's not, let me know. So I think one of the crucial parts about executive functions is that you understand these different steps, like in this entire cycle, what belongs to the executive functions and not. If we're talking about inhibition, it's mostly the inferior frontal gyrus that's taking care of inhibition. You said 31, right?
36, 38.
36, 38.
I can still see the question from the weekly assignment.
Yes, I can't see your screen either.
Okay.
Yeah, me too.
Sorry. I'm stopping the sharing and share again. That's this one. Now you can see it?
Yes.
Yes.
Okay, good. So I think if we're talking about the stop signal reaction time, I'm just going to go one slide earlier. So this is 35. And this is quite a tricky experiment that you can use to measure inhibition. So in most of these experiments, for example, when measuring this in macaques, then you ask the macaque, the monkey, to maintain fixation to the center of the screen, as you can see in the no stop signal trial. Then suddenly the square moves either to the right or the left, and the instruction is to make a response or an eye movement as fast as possible. You can also do a similar kind of task in humans, of course. You basically maintain fixation, and then the stimulus moves to the left or the right, and you have to make a response with your left or your right hand as fast as possible. So in the no stop signals, you basically, everything happens as planned, right? So the square moves to the right, and then you make a response with the right as fast as possible. In the stop signal trials, something nasty happens. So here again, the square moves to the right. You prepare to make a movement, but then a cancellation stimulus is shown in the center of the screen. It can also be a sound. So this is quite typical that you actually have the stimulus move to the left or the right. And then whenever you hear a beep, then you have to abort your response. In this case, in this paradigm, you manipulate the cancellation stimulus. So the stop signal delay, as you see it here. So for example, if I move the stimulus from the center to the right, and then immediately you hear the sound, you will always be able to cancel your response. That's trivially easy. If I play the sound 1 second after moving the stimulus, you have always made the response, so you're always incorrect. And we're actually trying to find out like how to best do this with the stop signal reaction time. And one of the things that what I try to explain here is, I think the key element that you have to know is that a stop signal reaction time that is high signals that you have low inhibition, and a stop signal reaction time that's low actually shows high inhibition. We're basically like, how much time can I leave between the go and the stop signal before you're actually before you actually, when you're still able to basically withhold your response in 50% of the cases. So the general thing that we're getting at here, so the stimulus moves to the right, the response generation process starts. So you're actually starting to prepare your motor cortex to make a response. A little bit later, you get a stop signal. It again needs to travel across the entire brain. And if it's slower, as in this slide, than the response generation process, you make a response. Because the stop signal arrives in the motor cortex too late. You already made the response. In this example here, you can see a similar kind of example. Here, the response inhibition process starts later than the response generation process, but it arrives, it basically finishes the race earlier, so you're able to withhold your motor response in this case. So that's basically what we're trying to get with this experiment. This stop signal reaction time is just a reaction time indication of how well you are, how good your response inhibition works. But the general idea here is that I have the response generation process, that's always the same. I basically jitter or vary the response inhibition process to see how much time I can leave in between these two processes. And of course, if you have a very good inhibition, then I can start the response inhibition process later in time, and you still have time to withhold your response. So conceptually, just remember, if the SSRT is high, you have low inhibition. If it's low, you have high inhibition. Any other questions about this?
Nope.
I asked like to explain this a couple of years ago to, I think, master students, and almost everybody failed. So I think it's unfair to ask any more questions on this. It's basically a race between two processes, a go process and an inhibition process. And this is just an indication. So this is a behavioral indication of the efficiency of inhibition. And that's the most important thing to remember. The SSRT tells you something about how well inhibition is implemented. Okay, can we go to the next question?
Yeah, let's have a look. Are there any other questions? Nope.
No?
No. Can you please go back to the FL, Serena?
What is the FL?
Yeah, I'm not sure. Executive functions.
Yeah, sure. I'm going back to executive functions.
Frontal lobe.
Frontal lobe. Which slide?
Serena. What type of task is this related to?
I think you might want to unmute yourself to explain it a bit more in detail.
Just so frontal lobe is responsible for more complicated cognitive tasks, but like which ones, and which ones that are complicated maybe are done in different areas.
Okay, I think it's so I don't know where you is the link to the video in there as well. Let me. Sorry, my computer is like extremely slow in loading this. So wow. I think what is a very important thing to state about the prefrontal cortex, and if we're talking about cognitive control or these executive functions, we're mostly talking about the lateral prefrontal cortex that you can see in red here. It's a very important thing to say. Like if we're talking about what is this brain region doing, you actually should be referring to the opposite question: what is it not involved to? So if we're talking about can you do math equations? Can you see things? Can you remember things? Can you have a conversation with someone else? You can do lots and lots and lots of things without a lateral prefrontal cortex. So that's something that is important to state here, because that's one of the surprising factors. This is something you wouldn't expect from this part of cortex. But what is very evident that any kind of goal-oriented behavior just is gone. So when you try to make a cup of coffee, you have to follow a sequence of actions, and that already needs some kind of supervision, some kind of ordering of all these actions to get stuff done. If you want to learn for your exam, you need to plan, and you basically need to break it up into separate sub-steps to actually make it happen. So all these kinds of goal-oriented behaviors are impossible without a dorsolateral prefrontal cortex or a lateral prefrontal cortex. So when it comes down to if you have multiple options to choose from, then you have a problem. So all kinds of convergent things. So if only one answer would be correct on, for example, most of the intelligence test batteries, then people with lateral prefrontal cortex damage still seem to be quite intelligent. When it comes down to like creativity, critical thinking, or problem solving problems, where you have to combine different rules and find more or new solutions, then you crucially need this lateral prefrontal cortex.
You mentioned,
So, yeah.
you mentioned something regarding conversations and lateral prefrontal cortex. I would imagine that even in conversations, people with damage to this area would still have limited conversations. I would imagine that even in conversations, you need working memory.
Yeah.
You need to.
Yeah, you're completely right. I think the thing is that what they don't have, these people, is working memory. So when you distract them just for 1 second, they lost track of what just happened. So they don't know anymore. But for example, if you have studied law or psychology and you ask, like you're learning for this exam, I asked you, like, can you give a summary of entire chapter 2 and 3? You would be able to do so, even if you don't have a lateral prefrontal cortex, because you store that in long-term memory. So in that sense, if I give you an instruction, you can still respond to this instruction with intelligent behavior. But if you want to keep track over time in the now, so to say, in a conversation, then you have a serious problem.
Okay, thank you.
I think I hope there's like I had a video, I think somewhere, a link to a video in this slide, and there they are actually showing a lawyer who had very extensive damage in the lateral prefrontal cortex, showing all kinds of intelligent behavior, but nothing goal oriented. So I think that's one important thing to. Like, let me see. Can I go to? Maybe it's no, it's not here. I think it's an excellent thing to, without working. I think it's this link. Let me just double check. If you haven't watched that movie, it's I think it's, especially if you like 80s hairdos, it's a great thing to see. Yeah, that's definitely the video. And there you see the patient what he cannot do anymore, and it's really quite impulsive. So there's no inhibition, there's no goals, there's no working memory. So they're kind of a slave to their environment. If you ask them something, they give an intelligent answer. If they see something in their environment, so for example, if you have a frontal lobe patient, they see a bed, they go lie in the bed. That's just basically their most acute immediate response to the environment. They act out on that. So there's actually no multiple choice anymore. There's no options available anymore. They cannot choose anything. They just do the thing that is most prominent. Like if there's an urge from their, say, emotional system, they act out.
Thank you.
Okay. We have 5 minutes left. Any more questions?
No, that was this for the chat.
Okay. If there's any more questions related to the procedure or something like that, I think I stated that on Canvas. The weekly assignment of this week only consists of 12 MCs and 1 open question. If you really want to retake it, then I need to grade your open question, then you can retake it, but it's the same questions over and over again. So there's really no use of taking it twice, I would say. they are really representative of what you can expect at the exam. We basically created all the exam questions, the re-exam questions, and the weekly assignment questions in one go. And then we picked some to put in the exam, some in the partial exam, some in the weekly assignment. So that's the kind of level that you can expect.
Okay.
I think that's just good to state here. I'm going to stop the share again. So I wish you good luck in getting like the final hours before the test is on, and I think you will be fine. And we probably will be hosting many more of these Q&As, hopefully maybe even like a weekly Q&A. So if anything is unclear, we stay on top of it.
Okay, that would be good.
Yeah, it's just a bit difficult with 2 kids at home running around, breaking down the house. So, and the 2 of us working. So that's the reason why we chose for this. But when they go back to daycare, it should be easier to do these things.
Okay, thank you.
Thank you.
Thanks. Yeah, and thank you, Claudia, for hosting this. I will download the chat and share that.
Yeah.
So all the questions and all the answers will be there.
Thank
Okay.
you.
Thank you.
Perfect. Thank you.
Thank you. Bye.
Bye.